I'm Courtney Siegert. I'm an associate professor in the Department of Forestry at Mississippi State University, and today I'm going to be talking about changes in bark hydrologic properties along the stem of 4 common southeastern tree species. I'd like to acknowledge my co-authors on this presentation, Kira Hampton, Amy Triplett, Lucas Ivanko, Brianna Ellis, Cade Booth, and Anna Elek. So, bark plays an important role in the forest hydrologic cycle, as it can absorb substantial quantities of water during rainfall events. Water can be intercepted and diverted into bark storage instead of being delivered to the forest floor where it would be available to plant roots. The quantity of water that can be stored in tree bark varies across species and is a function of bark surface properties and internal structural characteristics. For over 30 years, the role of bark in the forest hydrologic cycle has been documented. For instance, in Hurwitz in 1985 showed that bark surfaces represent 50 to 80% of canopy water storage of rainfall interception. More recently, Elek and colleagues have demonstrated the ability of bark to absorb moisture from the atmosphere during non-rainfall conditions, i.e. the property of hygroscopicity, and that the range of hygroscopicity across species represents anywhere from 10 to 60% of total bark water storage. This means that bark water storage may be partially full before rainfall events even occur. Bark structure varies as a function of tree species and along the tree stem. Structural characteristics can include bulk and specific density, actual hygroscopicity, potential hygroscopicity, which is hygroscopicity measured on ground bark samples to remove any influence of macrostructural characteristics, and also porosity. Across species, trends and properties of intact bark, including bulk density and actual hygroscopicity, have been shown to differ from trends and properties from ground bark, revealing differences in physical structures versus microstructures and bark chemical composition that influence water absorption and retention. For example, the chemical composition of cell wall macromolecules has been shown to affect how well natural fibers can absorb water vapor from the atmosphere, suggesting that similar mechanisms may exist in bark composition across species. We have preliminary evidence from conifers that indicates that these bark traits can vary along the length of the stem, but our understanding of this variability is limited, especially with respect to other components of the forest hydrologic cycle. To contribute to our understanding of the role of bark in the hydrologic cycle, we conducted an experiment to assess how bark physical and chemical properties vary across species and along tree stems in the southeastern United States. We selected 4 common tree species that are widely distributed in mesic areas in the southeastern United States, including loblolly pine, shagbark hickory, sweet gum, and cherry bark oak. In these top pictures, you can see the cross-sectional distribution of bark in loblolly pine here, with a very thin layer of inner bark, that's that lighter color, and the majority of the bark volume made up of outer bark with the darker color. In sweet gum, we have a more even distribution of inner bark to outer bark, and of shagbark hickory also has a relatively even distribution of inner versus outer bark, but with visually more dense tissues. In the picture below are examples of the 4 species included in our experiments with different external bark displayed, with loblolly pine, shagbark hickory, sweet gum, and cherry bark oak. At 3 different locations in the study area, 1 tree of each species was selected for sampling. Each tree was 16 to 20 centimeters in diameter at breast height. After felling, each tree was divided into 10 sections based on total height, and in total, 12 trees were sampled for this study. Bark samples were removed from each section using hammers and chisels for analysis. Due to difficulties in extracting very thin bark from upper section of the trees, only the lower 6 sections are described in our results. To assess differences in bark hydrologic properties as a function of bark macrostructure across species, we used intact whole bark samples and conducted the following experiments. First, we measured bulk density as the ratio of the mass of oven-dried bark sample to the volume of the bark that was determined via water displacement methods. The actual hygroscopicity experiment was done by placing dried bark samples in desiccators filled on the bottom with water to create 100% relative humidity conditions. The bark was not submerged in the water. Bark was measured every 1 to 2 days for 8 days, whereby the increase in mass is attributed to the absorption of water vapor from the 100% relative humidity environment. Prior to placement in desiccators, all surfaces of the bark, except the outer face that would naturally be exposed to the atmosphere in the forest, were sealed in silicone caulk. Moisture content was determined as the maximum mass of water absorbed as a percentage of the total dry mass of bark during the actual hygroscopicity experiment. And total porosity was measured as the volume of pore space within bark as a ratio of the difference between specific density and bulk density to specific density. To assess differences in bark hydrologic properties as a function of microstructure and chemical composition, we used bark samples that were ground to less than 0.25 millimeters and conducted the following experiments. First, we measured specific density as the ratio of the mass of ground oven-dried bark sample to the volume of bark determined via pincometer method using 100% ethanol. Then, potential hygroscopicity was measured in a method similar to actual hygroscopicity, where the ground bark samples were placed in a crucible inside the 100% relative humidity desiccator. We ran a mixed model analysis using the LMER function in R to understand how each property changed as a function of tree species, stem section, and the interaction between the two. Because there were 3 trees of each species and 3 bark samples from each stem section of each tree, tree and bark replicate were treated as random effects in the models. Results is bulk density. In all of the figures, the bark property is displayed on the x-axis. Here is bark bulk density, and the stem section increasing from the base of the tree upwards are plotted on the y-axis. Overall, loblolly pine bulk density was lowest at 0.33 grams per cubic centimeter, followed by sweet gum at 0.39 grams per cubic centimeter, hickory at 0.54 grams per cubic centimeter, and cherry bark oak at 0.59 grams per cubic centimeter. These trends were generally consistent along stem section. At the species level, cherry bark oak in blue, sweet gum in yellow, and loblolly pine in red bulk density varied along stem section, while hickory bulk density in gray did not change. Even though bulk density varied along stem sections, it did not trend consistently in one direction or another, i.e., it did not consistently increase with stem height or consistently decrease with stem height. Actual hygroscopicity was not significantly different among species or along stem section, but was significantly different considering the interaction between species and stem section. Sweet gum actual hygroscopicity in yellow varied as a function of stem section, while the other 3 species did not change. Moisture content varied across species, with loblolly pine bark in red having the greatest maximum moisture content at 22%, followed by sweet gum in yellow at 17%, hickory in gray at 16%, and cherry bark oak in blue at 14%. Across stem sections, moisture content varied in cherry bark oak, but not in the other 3 species. Porosity varied across species and stem section. Cherry bark oak in blue had the lowest total porosity at 63%, followed by hickory in gray at 67%, sweet gum in yellow at 70%, and loblolly pine in red at 76%. Total porosity varied along stem section in cherry bark oak, in sweet gum, and in hickory, but not in loblolly pine. Specific density varied across species, with ground hickory bark in gray having the highest specific density at 1.66 grams per cubic centimeter, followed by cherry bark oak in blue at 1.16, loblolly pine in red at 1.43, and sweet gum in yellow at 1.32 grams per cubic centimeter. Specific density variability along stem section was large, and there were no differences in height within a given species. Finally, potential hygroscopicity of ground bark was greatest in loblolly pine in red at 5.7 millimeters of water per centimeter of bark thickness, followed by sweet gum in yellow at 4.2 hickory in gray at 3.24, and cherry bark oak in blue at 2.43 millimeters of water per centimeter of bark thickness. Loblolly pine potential hygroscopicity increased with tree height. Potential hygroscopicity also varied with height along stems in sweet gum, hickory, and cherry bark, but not in a consistent direction as they did with loblolly pine. In summary, we observed more variability among species than along tree stems within a given species. All bark traits except actual hygroscopicity varied across species. Bark density, total porosity, and potential hygroscopicity varied along stem sections, but not consistently in one direction with height. As shown in this principal components figure, bark density, specific density, and actual hygroscopicity were strongly correlated, where porosity, moisture content were strongly correlated with potential hygroscopicity. Taken together, results suggest that the role of bark in the forest hydrologic cycle is diverse and complex across species and vertically along the tree stem. Our current understanding of bark as a reservoir of water storage and our parameterization of bark in hydrologic models is pedestrian and deserving of further investigation. With that, I'll take any questions. I've listed my email address and my Twitter handle that you can reach out to me at. Thank you.